Good day, everyone. Today, we will explore one of the most essential tissue types in the human body, the epithelial tissues. These tissues line our body surfaces, cover our organs, and form many glands. They serve crucial roles such as protection, absorption, secretion, and sensation. Epithelial tissues are also among the easiest and most important structures to identify under the microscope because their appearance, whether flat, cuboidal, columnar, simple, or stratified, tells us a lot about their function. In our laboratory activity, we examined several epithelial tissue slides, each showing different characteristics and adaptations. Before discussing our observations, let us first review the materials, setup, and procedures used in viewing these epithelial samples. For the procedures, the following steps were performed during the activity. First, the slides were arranged and labeled properly before viewing. Begin examining each slide during the low-power objective to get a general view of the specimen. Once the overall field was visible, use the fine adjustment knob to bring the tissue into sharper focus. After establishing a clear image, shift to higher magnification to observe more specific details such as cell shapes, layering, and distinct structural features. For every slide viewed, record the notable characteristics that were present. After completing all observations, compare the findings from each slide to identify similarities and differences between the various epithelial tissue types.
And for the importance, studying epithelial tissues is important because first, they form protective barriers that guard the body against injury, dehydration, and pathogens. Second is they reflect organ function, and their structure provides insights into absorption, secretion, or protection. Third is many diseases starts in epithelial cells, including most cancers, making histological identification essential. And lastly, laboratory diagnosis depends on recognizing epithelial patterns, especially in biopsies, smears, and tissue sections. Next is the human brown skin section. This showed stratified squamous epithelium with keratinized layers at the surface. The pigment melanin was visible in the deeper layers, giving the tissue its brown color. This represents epidermis of pigmented skin, especially in areas like arms, back, or face where melanin is abundant. This structure protects against UV radiation, dehydration, and mechanical injury. For our next one, it's the human scalp vertical section or VS. The epidermis displayed stratified squamous keratinized epithelium. It has visible hair follicles, sebaceous glands, and layers of the skin were noted. Located at the scalp region of the head, specifically areas containing hair follicles. The hair follicle is a modification of epithelium, showing how epithelial tissues produce appendages for protection and sensation. For our fourth one, it's the lacrimal gland section. Tissue showed simple cuboidal to low columnar epithelial cells arranged in clusters or acini. Light staining cytoplasm indicated active secretion, and it can be found at the lacrimal gland located in the upper lateral portion of the orbit near the outer corner of the eye. And this type of epithelium specializes in secretion, producing tears that lubricate and protect the eye. And lastly, the human palm skin section. Thick stratified squamous keratinized epithelium, noticeably thicker than normal skin. Clear stratum lucidum layer was visible, a hallmark of thick skin. It is located at the palm of the hand, classified as thick skin, and the thick epithelial layer provides protection against friction, pressure, and repeated mechanical stress. And for the importance, studying epithelial tissues is important because first, they form protective barriers that guard the body against injury, dehydration, and pathogens. Second is they reflect organ function, and their structure provides insights into absorption, secretion, or protection. Third is many diseases starts in epithelial cells, including most cancers, making histological identification essential. And lastly, laboratory diagnosis depends on recognizing epithelial patterns, especially in biopsies, smears, and tissue sections.
Next, the tissue that we will discuss is the connective tissue. It is one of the four primary types of tissues in the human body, alongside epithelial, muscle, and nervous tissues. Unlike other tissues, connective tissue is characterized by an abundance of extracellular matrix, which is the non-living material that surrounds its cell and is composed of protein fibers and ground substance. This unique features allows connective tissue to provide support, strength, and elasticity to different parts of the body. Connective tissues serve a wide range of functions. They connect and hold organs and tissues together, protect organs, store energy, and even play a role in transporting substances through the body, as seen in the blood. Before we start for the observation, let us first review the materials, setup, and procedures used in viewing these epithelial samples. For this microscopic observation, the following materials were used. Microscope. Next are the prepared slides. These were the steps performed during the activity. Slides were arranged and labeled properly before viewing. Each slide was examined, starting with the low power objective. Once the general field was seen, the tissue was brought into sharper focus using the fine adjustment. Higher magnification was used to identify cell shapes, layering, and specific structural features. Notable characteristics were recorded for each slide. After all observations, comparisons were made between the different connective tissue types.
So, moving on, here are the observations from each connective slide examined. So, first, human tendon. Tendons are dense regular connective tissues made mostly of collagen fibers arranged in parallel bundles. Tendons connect muscles to bones, transmitting the force generated by muscle contraction to produce movement. Their dense collagen provides high tensile strength to resist stretching. Next is the mammary gland section. Mammary glands are compound tubuloalveolar glands embedded in connective tissue. They produce and secrete milk for infant nourishment. The connective tissue supports the glandular structure and aids in the distribution of blood vessels and nerves. Human umbilical cord. The umbilical cord contains Wharton's jelly, a loose connective tissue rich in mucopolysaccharides surrounding two arteries and one vein. The fibroblast-like cells, nuclei, are scattered throughout the matrix. It provides structural support and protection to the umbilical blood vessels, allowing nutrients and gas exchange between mother and the fetus. The reticular tissue is a loose connective tissue with a network of reticular fibers, or also known as type 3 collagen. Nuclei of reticular cells are located along the fibers. It forms the structural framework or stroma of soft organs like spleen, liver, and lymph nodes, supporting blood cells and immune functions. Lastly, human scalp. The scalp cross-section shows layers of dense, irregular connective tissues in the dermis, with fibroblast nuclei scattered among thick collagen bundles. It provides mechanical strength and elasticity to the scalp, anchors hair follicles, and supports blood vessels and nerves for skin nourishment and sensation. Studying connective tissue is important because they provide the body with essential support, protection, and structure, forming framework that holds organ in place and shield vital areas like the brain and heart. They also connect different tissues and organs with tendons and ligaments, enabling coordinated movement. Blood is specialized connective tissue, transports oxygen, nutrients, hormones, and waste production to maintain the body's internal balance or homeostasis. Adipose tissue stores energy in the form of fat and helps regulate body temperature through insulation. In addition, connective tissues plays a major role in repairing damaged areas and supporting immune responses that protect the body from infections. That's all. Thank you for listening.